In our previous video, we discussed how NAMPT is controlled by the circadian clock. In this video, we will discuss the complex relationship between metabolism, the circadian clock, and NAMPT. NAMPT encodes nicotinamide phosphoribosyltransferase, an enzyme that is critical for the production of NAD+. NAD+ is both an electron carrier essential for cells to transfer chemical energy and also a cofactor for enzymes that modify gene expression or repair damaged DNA. NAD acts as an electron shuttle for various reactions including fatty acid oxidation, the Krebs cycle, and glycolysis. These processes use interlinked reactions that ultimately use NAD+ to carry chemical energy used to make ATP. NAMPT converts one of the building blocks of NAD+, called NAM, into NMN. NMN is then converted into NAD+. As NAD+ is used in reactions, it is converted back into NAM, and NAMPT is needed to generate more NAD+. NAD+ activates a protein called SIRT1. SIRT1 binds to the clock BML1 dimer and regulates the molecular clock. Thus, NAMPT activity affects the levels of NAD+, which affects the circadian clock, and the clock controls the production of NAMPT. This feedback connects the circadian clock to metabolism. As we discussed in our previous video, when the circadian clock is disrupted, NAMPT expression decreases and becomes arrhythmic. What are the consequences of low NAMPT expression? Well, mice with mutations that remove NAMPT entirely are embryonic lethal. This means that NAMPT is so important to survival that the NAMPT knockout animals don't live long enough to be born. Instead, researchers can examine the role of NAMPT by making conditional knockouts, meaning that NAMPT is not missing in all cells all the time. Rather, loss of function can be specifically targeted to a tissue or developmental stage. In many cases, animals can survive an embryonic lethal gene being knocked out in certain tissues or later in development. To engineer mice for conditional loss of a gene, the Cre-Lock system is used. This approach uses Cre, a site-specific recombinase, and LOX B sites, sequences of DNA recognized by Cre. Scientists engineer Cre to be expressed only in a particular tissue or under specific conditions. For our example, let's link the Cre gene to the promoter of the gene that codes for albumin. Like all of our genes, the genetic code for albumin is stored in the DNA of every cell, but albumin is typically only expressed in the liver. If we insert the Cre gene under the control of the albumin promoter, then although the Cre gene will be in the DNA of every cell, the Cre protein will be made only in the tissues where albumin is made. When a cell transcribes the albumin gene, it will also turn on the Cre gene, and cells that do not synthesize albumin, like in the kidney, will not make the recombinase. LOX B sites are placed around an important part, such as the critical exon of another gene. Let's say NAMPT. When Cre recombinase is expressed in cells that make albumin, Cre recognizes the LOX B sites and excises the NAMPT gene sequence. Thus, these specific cells will not be able to express functional NAMPT, while other cells are still able to. Using Cre-Lock's technology, scientists found that when NAMPT production is knocked out in skeletal muscle, it causes muscle weakness. This is believed to be caused by an observed decline in ATP content and NAD+ in the mitochondria. Loss of NAMPT in projection neurons, which link the brain to the muscles, leads to neurodegeneration, causing motor abnormalities, muscle atrophy, and eventually death. Deletion of NAMPT in adipose tissues causes severe insulin resistance. Finally, loss of NAMPT in pancreatic beta cells leads to a decrease in the ATP-ADP ratio and disrupts the insulin excretion signal. Having no NAMPT is problematic, but what about the low, non-rhythmic NAMPT seen in circadian mutant mice? Let's look at the role of NAMPT in mitochondria. When an animal fasts, it oxidizes fat to form ketones and produces ATP in a process called fatty acid oxidation. In mitochondria from the liver of mice that lack the core clock gene BML1, scientists found a decrease in both NAD+ and activity of the oxidative pathways that carry out fatty acid oxidation. BML1 knockout mice die after relatively short periods of food restriction compared to wild-type animals. The sensitivity of the BML1 knockouts is believed to be due to a decrease in the ability to oxidize their fat for energy. The scientists hypothesized that this decrease in fatty acid oxidation and NAD+ is due to a decrease in NAMPT activity, so they gave the circadian mutant animals NMN to bypass NAMPT. Once they did this, the liver mitochondria in the circadian mutants had normal NAD+ levels and recovered fatty acid oxidation activity in these mice. These experiments show that the circadian clock controls mitochondrial oxidative metabolism by regulating the NAMPT-dependent productions of NAD+. So let's review what we've learned. The circadian clock controls NAMPT production. NAMPT production feeds back to the clock through NAD+ and SIRT1. NAMPT production is critical for cellular metabolism, and some of the negative metabolic effects of disrupted circadian rhythms occur because of decreased levels of NAMPT. These feedback processes are critical for survival and reveal the interdependence of circadian timing and metabolic pathways.